Our presentation is going to focus on techniques used to detect prion infections. We found that detection techniques are used post-mortem and could be improved since there is an increase in infection cases, but a deficit of effective techniques. This means that if these prions are detected, it is usually after the animal has passed from the infection and could have possibly spread to a much greater population. With that being said, we are going to analyze 3 post-mortem prion detection techniques and suggest different methods on how prion detection techniques can be improved. One technique that is currently being used is biological assays through tissue and blood samples, meaning that DNA is gathered via blood or oral sample and is analyzed through desired bioassay. Henderson et al. Showed that this technique is minimally invasive, which is ideal for management plans. This technique has also shown potential to detect prions in low concentrations, which is beneficial because this infection is usually found in low concentrations. Studies showed that using saliva samples with RT-Quick assays provided the detection of prions with a dilution ratio of 1 to 10 in saliva samples and the increased sensitivity of detection in deer saliva samples, which would allow for higher detectability. One con with this technique is that it only amplifies for a single round of DNA. Henderson et al. Explains that since these prions are found in such low concentrations, they may require multiple rounds of amplification, leading to a large amount of work. McNulty et al. Explained that this technique can be costly and that bioassays do not directly measure the infectivity since their studies showed that a limitation of this technique is the direct measurement of infectivity due to the insufficient quantities of brain tissue negated. And this figure shows the practice of saliva sampling, in this case with a dog. The next technique we will be talking about is the real-time quaking-induced conversion, or RT-Quick assays of third eyelids and cervids. Cooper et al. Explains that third eyelids are found between the globe of the eye and the lower eyelid and are easy to access. This study also showed that this technique held a 4.5% for false positives, which solidifies that this technique has a high consistency to test for prion detection and detects prions as early as 1 month after inoculation in deer. Early detection of prions is important because it could help determine where infected animals came into contact with other individuals and decrease the spread of infection. One setback of this technique is the inability for early detection in all species tested. Cooper et al. Shows that the RT-Quick assay in the third eyelid of pre-symptomatic 396GS genotype deer did not show detection. And this figure shows the detection of prions in third eyelids from the study that was mentioned in the last slide.
The final technique that we have analyzed is the evaluation of immunoassays in sheep to test for scrapie in fetuses. Scrapie is a transmissible spongiform prion infection present in sheep, just as chronic wasting disease is in cervids. A protein misfolding cyclic amplification technique, PMCA, has allowed prion detection in different tissues and excretions that the misfolded protein has been undetected by in traditional assays. Due to the similarity of prion infection, these findings may be translated to prion infections of wild populations. The researchers' in vitro technique is the first to show lateral transmission of infection. However, Garza et al. Did explain that drawbacks of this technique are that this testing does not elicit how the transmission from mother to offspring occurs, and it requires extreme amplification for protein detection, which can be quite costly and time-consuming. Figure 4 shows the differences between a control brain and a brain and saliva sample that have scrapie, with emphasis on the PRP protein on the far right. Figure 5 is a diagram of the bioassay technology that shows how the immunoassay evaluation is completed. Each of the techniques that we have reviewed are all post-mortem and cannot provide high detection rates. Although detection of the prion in the first place is extremely important, high detection conducted ante-mortem would allow for intervention before an entire population was infected. These prion infections are so highly contagious that by the time individuals in a population die, many members of the population are already infected. Pre-mortem detection methods could allow for culling of infected individuals to prevent spread. The good news is, better methods are on the way. There is promising research by veterinary microbiologists in antemortem detection by rectoanal mucosa-associated lymphoid tissue, or RAMALT, and nasal brush samples. These samples are currently only collected from farmed white-tailed deer, but this method has the potential to be used on wild populations in the future. This technique is both antemortem and produced the highest detection rate of 69.8%. Haley and the rest of their research team explained that future research in this area has potential for large-scale automated testing of chronic wasting disease. Given the techniques we have evaluated, this seems like the best measure for detecting prion infections successfully and as early as possible. This figure shows nasal brush samples being collected from farmed white-tailed deer. Thank you so much for watching our video, and we hope that we've provided a little bit of insight into the world of prion detection. If you have any questions or comments, please post them in the comment section below.